These our RNA molecules with gel, that is our cubes to be added by gel plus our RNA molecule plus as a formaldehyde solution with buffer solution. So we are start by our electrophoresis apparatus. So this this gel with RNA solution, they are move in upward direction. So they are move. We are used by specific types of the membrane. This membrane is called as a nylon membrane or nitrocellulose membrane. Also we are used by Watts paper, okay, Wattsman's paper, and these are very important to be separation of the RNA molecule. So that is our nylon membrane. In this membrane, gene of interest are tightly attached. So after separation, we observe many bands. Okay. So after separation, we are separated by our nylon membrane. So look at this nylon membrane. That is the our gene of interest. So this gene of interest, this nylon membrane, we are treated with as a UV light, because very important to be this RNA molecules are tightly attached with the membrane. Okay. So we are treated with as a UV light solution. Okay. Then this membrane we are add by prop solution. For example, this membrane we are add to be prop solution. That is our nylon membrane, and this is our prop solution. So guys, what is the prop solution? So prop solution is a complementary sequence of our gene of interest. Complementary sequence of gene of interest. What is the complementary sequence? They are the short oligonucleotides, such as 100 to 150 short oligonucleotides chain. Okay, they are made by one of the RNA solution. Okay, and this prop solution. So guys, we are already known their sequences. So this complementary sequence are carry binding with our original molecules, and after they bind, we look at some types of bands. So this complementary sequence are carry to be binding to each other. Okay, they are hybrid to be each other. So this sequence we are called as our gene of interest. That is a very beneficial technique to be easily separated by our RNA molecules. We want to be any cell, any cell or any other solutions to be want to be separated RNA molecules, DNA synthesized by them, and want to be known their sequences, known their complementary sequences in any cases. So we are applied to be always in northern blotting techniques. It is very beneficial and very applied to be quickly separation of these molecules on basis of molecular size. I hope guys you are understand to be second blotting technique as a northern blotting, that is the very important for the separation. Okay, so take by screenshot, and thank you so much.